How does epigenetics influence bipolar disorder? Imagine your genes are like a set of instructions for your body, but epigenetics acts like a dimmer switch that can turn these instructions up or down without changing the actual words. This means that environmental factors such as stress, trauma, or even medication can influence how your genes are expressed. In bipolar disorder, this epigenetic regulation plays a key role in how the condition develops and varies over time. Epigenetics involves several mechanisms, including DNA methylation, where chemical groups are added to DNA, often reducing gene activity. Histone modifications, like methylation and acetylation, change how tightly DNA is wound around proteins, affecting gene accessibility. Non-coding RNAs can also regulate gene expression by interfering with messenger RNA. These processes can alter the activity of genes involved in mood regulation, neural plasticity, and how the brain responds to stress, all of which are important in bipolar disorder. While genetics provide a foundation for bipolar disorder, they do not tell the whole story. Environmental influences can modify gene activity through epigenetic changes, which helps explain why two people with similar genetic backgrounds might experience different symptoms or disease courses. For example, changes in DNA methylation have been observed in genes related to neurotransmitter systems and neurotrophic factors, which are essential for healthy brain function and mood stability. One well-studied gene affected by epigenetic changes in bipolar disorder is the brain-derived neurotrophic factor gene. This gene supports the growth and survival of neurons, and its activity is regulated by epigenetic modifications. During depressive episodes, patients often show increased methylation of the BDNF gene, which reduces its expression. This decrease can impair neural plasticity and contribute to mood dysregulation. Conversely, during manic episodes, methylation levels tend to be lower, showing that epigenetic changes can be dynamic and linked to mood states. Other genes, such as those encoding potassium channels, also show altered methylation patterns in bipolar disorder. This suggests that epigenetic dysregulation affects how neurons fire and communicate, influencing mood and behavior. Interestingly, studies have found differences in epigenetic marks even in identical twins, where only one twin has bipolar disorder, highlighting how environment and epigenetics interact beyond just inherited DNA. Medications like lithium, a common mood stabilizer, may work partly by influencing the epigenome. Lithium has been shown to affect DNA methylation and histone modifications, helping to restore normal gene activity. This supports the idea that epigenetic changes are not just involved in causing bipolar disorder but could also be targets for future treatments or markers for how well someone might respond to therapy. In essence, epigenetics acts as a bridge between your genetic makeup and your life experiences. It helps explain why bipolar disorder can look different from person to person and why environmental factors matter so much. Researchers are actively exploring specific epigenetic markers that could someday help with diagnosis or personalized treatment plans. But it is important to remember that this field is still evolving, and more studies are needed to fully understand these complex processes. Always consult healthcare professionals for personalized advice, as bipolar disorder involves many factors, and each person's experience is unique.